Hello students, welcome to SWAM course on microbial technology. Topic we discuss here is pathogenic microbes. I am Soumya Khare from Chhattisgarh Swami Vivekanand Technical University, Durg. Previous lecture we studied on microbial diversity, here which we describe the characteristics used to classify bacteria and viruses specified with DNA and RNA. Explain a specific property of viruses that distinguish them from bacteria. Define the term diplococci, streptococci, staphylococci, coccobacillus, diplobacillus, streptobacillus and polymorphism. Identify several important bacterial diseases of human, and also we state several ways in which archaea different from bacteria. Topic to be covered in this lecture on pathogenic microbes. We learn about some of the common features that microbes organism must have in order to be infectious, explore the wide variety of organisms that are known to cause disease in human. Student will able to describe pathogenicity mechanism of infectious diseases. Here we know the content. We start with introduction, different types of pathogenic microbes, source of pathogenic microbes, pathogenic property of bacteria, pathogenic property of protozoa, pathogenic property of fungi, and microbial mechanism of pathogenicity, sign and symptoms of pathogenic microbes, identification, treatments, prevention measure, summary, and then last we conclude with some references. Now start with introduction. Pathogenic organism. What does pathogen means? It breaks down the word into two roots. First, patho, Greek means patho meaning suffering or disease, and gene, Greek word gene means to be born or produce. Pathogens are definition organism that can cause disease are known as pathogens. The severity of the disease caused by various pathogens varies greatly as well. In a hostile sense, pathogens are foreign intruder that target our body. However, the basic goal of a parasite or pathogen is to survive and reproduce just like any other organism. The definition of a pathogenic organism is an organism capable of causing disease in its host. A human pathogen is capable of causing illness in human. There are different types of pathogenic microbes. A pathogens bring disease to its host cell. Another name of pathogen is an infectious agent. They cause infection. As with many microorganisms, pathogens prioritize survival and reproduction. The human body immune system acts as a defense against pathogens. The body can easily fight off some pathogens, but others are potentially fatal. A pathogen infects its host and causes illness. Since pathogen causes infection, another term for them is infectious agent. Pathogens emphasize reproduction and survival, much like many other organisms. The immune system of human serve as a barrier against infection. Certain infections are easily defeated by the body, but others have the potential to be lethal. Various pathogenic microbes. The variety of microorganisms can cause diseases. Pathogenic organisms are 5 main types: viruses, bacteria, fungi, protozoa and worms. In this image, we can see that infectious disease are caused by pathogens which include bacteria, fungi, protozoa, worms, viruses. The pathogen of all classes must have mechanism for entering their host and invading immediate destruction by the host immune system. Source of pathogenic microbes. Bacteria are commonly found in various environments such as animal, food, human, air, water, soil and plants. Pathogens can spread in a variety of different ways. For example, coughing or sneezing can cause pathogens to spread throughout the tiny droplet in air. This droplet can contain microorganism which other people breathe it. Microorganism can also travel straight into the gut when a person consumes contaminated food or water. Bees, also infected insects, can also spread diseases. Pathogenic property of bacteria. Bacteria are microscopic pathogens that reproduce rapidly after entering the body. They can release toxin that damage tissue and causes illness. Bacteria are the most abundant microorganism present almost everywhere. They can produce toxin or elicit a strong immune response to damage host cells. Some of the important characteristics of bacteria are they are free living as well as parasites. They are autotrophs as well as heterotrophs. The bacterial cell membrane is made up of phospholipid. The cell wall is made up of peptidoglycan. Peptidoglycan is a combination of polysaccharides and amino acids. Bacteria mainly reproduce by binary fission. It is a kind of asexual reproduction. Bacteria also show primitive types of sexual reproduction, though through direct DNA transfer from one bacterium to another by the process of conjugation, transformation or transduction. Transduction process is the virus mediated and it causes the disease. Bacteria have prokaryotic cells. They lack a nucleus and other membrane enclosed organelles. Bacteria are classified as gram positive and gram negative based on their reaction to the gram stain and the structure of the cell envelope. Not all bacteria are pathogenic, though in the body there are many types of harmless bacteria and some may even support essential bodily function. These are the diseases by bacteria in human. The name of the bacteria and the disease caused by them. Clostridium typhni is called tetanus. Salmonella typhni is called typhoid. Mycobacterium tuberculosis, disease caused tuberculosis. Corynebacterium diphtheria called diphtheria. Streptococcus pneumoniae called pneumonia. Mycobacterium leprosis called leprosis. And Pandouma pallidum called syphilis. These are the bacterial diseases which is very infectious to the human body. Now, the pathogenic property of viruses. Viruses are the smaller than bacteria. A virus invades a host cell. It can replicate, producing hundreds and thousands of new viruses that go on to infect more host cells. Viruses are not considered as a living organism. They can only replicate within the host. They contain DNA or RNA as a genome which is enclosed within a protein coat. They are very small in size and the size of the virus range from 20 to 300 nanometer. Some of the important characteristics of viruses are as follow. Viruses are obligate parasites. They are inactive outside the host cell, but it is more active inside the host cell. The genetic material is either DNA or RNA. The genome may be circular or linear. The genetic material could be single or double stranded RNA or DNA. Generally, plants are infected with single strand RNA viruses and animals are infected with single or double strand RNA or double strand DNA viruses. The genetic material is present inside the protein coat are called capsid. Many viruses have an outside envelope made up of lipids. Viruses that infect bacteria are called bacteriophage. Viruses can pass from person to person in various ways, including via respiratory droplet that travel through the air, through contact with the blood of a person with the infection, through the contact with the body fluid of some with the infection. Now, the disease caused by viruses in human. The name of the viruses and the disease caused by them are given below. Vibrio minor and Vibrio major is causes disease caused smallpox. Rhinoviruses disease cause common cold. Mumps viruses disease cause mumps. Smilex viruses disease called herpes. Influenza viruses A, B, C, D are the variety of influenza viruses called influenza. Severe acute respiratory syndrome coronavirus 2 disease cause COVID-19. Human immunodeficiency viruses disease cause AIDS, and polio viruses disease cause polio. Pathogenic property of protozoan. Protozoa are unicellular eukaryotic. They lack a cell wall like animal. They are sometimes referred to as one-celled animal. They are heterotrophs. They are either free living or parasite. They cause various diseases in human. Some of the important characteristics of protozoa are as follow. They are single cell eukaryotes. The size range from 1 micrometer to several millimeter. They are heterotrophic. They show phagocytosis and pinocytosis. They have various organs for locomotion such as flagella, pseudopodia, cilia. On the basis of their locomotory organ, they are classified as flagellates, amoeba, sporozoa and ciliates. Amoeba have also a pseudopodia, and sporozoa spore producing and lack of locomotory organs. They reproduce by both sexual and asexual means. Protozoa are responsible for most protist disease. Protozoa are single cell microorganisms that feed on other microorganisms, organic tissues and debris. The disease caused by protozoa in human, the listing are given below. Trypanosoma bruchis disease cause African sleeping sickness. Plasmodium species disease cause malaria. Leishmania disease called leishmaniasis. Entamoeba histolytica anabasis, and Trypanosoma cruzi called crack diseases. Now, the pathogenic property of fungi. Fungi are the group of eukaryotic organisms. They are heterotrophs. They are mostly sporocytes that absorb organic matter from dead and decay substrates. Parasitic fungi derive nutrients from living plants and animal. They have a cell wall made up of chitin. Some of the common features of fungi are they are found in warm and humid places. Fungi are free living or parasites. They are found in soil, air and water. Fungi are mostly filamentous. The filamentous body is called hyphae, and the mycelium is a network of hyphae. Some hyphae are aseptous and contain many nuclei, are known as hyphae. The fungal cell wall is made up of chitin and other polysaccharides. Vegetative reproduction takes place by fragmentation, budding and fission. They reproduce asexually or sexually by spore formation. Conidia, sporogeospores and zoospore are asexual spores. Euspore, acrospore, basidospores are the sexual spores. There are thousands of species of fungi, some of which cause disease in human. Common fungal skin condition includes athlete's foot and ringworm. These conditions are contagious and can spread through person to person when it contact. The disease caused by fungi in human. The name of the fungi and the disease caused by them are the given below. Canidia albicans called candidiasis, Cryptococcus neoform called cryptococcal meningitis, Epidermophyton called ringworm, Histoplasma capsulatum called histoplasmosis, Mucormycetes called mucormycetitis, Aspergillus disease caused aspergillosis. Now, we learned about the microbial mechanism of pathogenicity. Pathogens, many microorganisms that has the capacity to cause disease. Pathogenic microorganisms have a special property that allow them to invade the human body or produce toxin. Pathogenicity is the ability of a pathogens to produce a disease by overcoming the defense of the host cell. Now, the mechanism include the molecular mechanism of pathogenicity. First is the portal of entry, next is penetrate the host defense, next damage to the host cell, next portal of exit. Again, the virulence is the degree of pathogenicity, providing a qualitative measure of the pathogenicity or the likelihood of causing diseases. The key concept of molecular mechanism of pathogenicity have the several factors are required for a microbe to cause disease. After entering the host, most pathogens adhere to the host tissue, penetrate or invade host defense, and damage host tissues. Pathogens usually leave the body via a specific portal of exit, which are generally the same site where they enter initially. The first step of pathogenicity mechanism is portal of entry, the specific route by which a particular pathogen gain access to the body is skin. Most microorganisms cannot penetrate intact skin, enter hair follicles or sweat gland, sweat duct. Certain fungi, dermatophytes, grow on skin and produce enzyme that break down keratin. Mucous membrane and respiratory tract is the second most entry and the common portal of entry. Microorganisms are that are inhaled with droplet of moistures and dust particles gain access to the respiratory tract. Gastrointestinal tract microorganisms enter the gastrointestinal tract via food, water and contaminated finger. Genitourinary tract causes the conjunctiva. Third, parenteral route. Microbes are deposited directly to the tissue beneath the skin or mucous membrane, bites, injection and other wound. The preferred portal of entry, many microorganisms can cause infection only when they gain access through their specific portal of entry. Second step is the pathogenicity adherence. The colonization, adhesion and are the most important property of adherence. Colonization is the first stage of microbial infection, the establishment of a pathogens at the appropriate portal of entry. Adherence is also terminology as a attachment, is often as essential step in bacterial pathogenesis or infection required for colonizing a new host. Adhesion, process by which microorganisms attach themselves to cells, require that participation of a two factor. The first is receptor and the second is ligand. Ligand is a small molecule which bind to the receptor. Microbial adherence to a eukaryotic cell or tissue surface involve complementary chemical interaction between the host cell or tissue surface and the bacterial surface. Third is ligand and adhesin, the surface molecule or pathogens that bind specifically to host cell surface molecule. The adenosine of bacterial cells are chemical component of capsules, cell wall, specifically lipopolysaccharide, glycocalyx or pile, viral capsid or component of the envelope. Receptor, surface molecule on host tissue to which pathogens adhesin bind. These receptors are present on the host cell surface. The host receptor are usually glycoprotein located on the cell membrane or tissue surface. Mucus, the mucopolysaccharides layer of glycoaminoglycan covering animal cell mucosal surface. Interaction of ligand with host receptor can determine a specificity for host cell. Inability to make attachment protein or adhesion render the microorganism avirulent. Ability to change or block the ligand or its receptor can prevent infection. How bacterial pathogen penetrate host defense? Capsule cell wall component are the most important penetration of pathogens. The first we learn about the capsule. Some pathogens have capsules that prevent them from engulfment by phagocytosis, Streptococcus pneumoniae, Haemophilus influenza and Bacillus anthracis. The cell wall component protein in the cell wall can facilitate adherence or prevent a pathogens from being phagocytosed. M protein resist phagocytosis, Streptococcus pyogenes protein inhibit T helper cells and resist digestion Mycobacterium tuberculosis. Enzyme leukocidine destroy neutrophils and macrophage. Coagulants cause fibrin clot. Kinase destroy blood clot. Hydrolyase which destroy a mucopolysaccharide that holds cell together. Collagenase hydrolyze connective tissue collagen. Immunoglobulin A proteases destroy immunoglobulin A antibody. Fourth step is the penetration into host cell invasion. Protein expressed on the cell surface of various pathogens that alter actin filament of host cell cytoskeleton, allowing microbes to enter cells. Salmonella typhimurium causes the actin of the host cell cytoskeleton to form a basket that carries the bacteria into the cell. This is also known as cell membrane refilling. How bacterial pathogen damage the host cell? Using the host nutrients cytospore, in the Greek it is also known as iron carrier, are small high affinity iron chelating compound secreted by microorganisms such as bacteria and fungi. Direct damage. The host cell can be destroyed when pathogen metabolize and multiply inside the host cell. It disturbs the host cell function and produce waste product. Most bacterial damage is carried out by toxin. Microbial toxin production. Toxin produced by microorganisms including bacteria and fungi. Microbial toxin promote infection and disease by directly damaging host tissue and by disabling the immune system. It is a small molecule, peptide or protein that are capable of causing disease on contact with or absorption by human body या body tissue. Toxigenesis or the ability to produce toxin is an underlying mechanism by which many bacterial pathogens produce diseases. Toxin are the two types, endotoxin and exotoxin. Endotoxin are cell associated substances that are structural component of bacteria. Most endotoxin are located in the cell envelope. Endotoxin refer specifically to the lipopolysaccharides and lipooligosaccharides located in the outer membrane of gram-negative bacteria. Here the immune response of bacterial endotoxin. The endotoxins are divided into two components in the large and small amount. Small amount endotoxin targeted Kupffer cells, neutrophils, B lymphocytes and complement. The targeted Kupffer cells act on the increasing interleukin and tumor necrosis factor which causes fever. Neutrophils act on the canine which causes vasodilation. B lymphocytes activate and increase the antibody and complement activity increases inflammation. Endotoxin in a large amount divided into two which causes shock and intravascular coagulation. Now next toxin is exotoxin. Exotoxin are usually secreted by bacteria that act as a site removed from bacterial growth. However, in some cases exotoxin are usually released by the lysis of bacterial cell. Exotoxin are usually protein, minimum polypeptide that act enzymatically or through direct action with the host cell and stimulate a variety of host response. Most exotoxin act at tissue site remote from bacterial origin point of bacterial invasion or growth. However, some bacterial exotoxin act at the site of pathogens colonization and may play a role in invasion. This is the image of immune response of bacterial exotoxin. This figure shows that exotoxin are secreted by bacterial cell. The Clostridium botulinum are the example that are toxic to somatic cells. Somatic cells have antibody on the cell wall to target exotoxin and bind to them, preventing the invasion of somatic cells. The binding of the exotoxin and antibody form an antigen antibody interaction, and exotoxins are targeted for the destruction by the immune system. If this interaction does not happen, the exotoxin bind to the exotoxin receptor that are on the cell surface and causes death of the host cell by inhibiting the protein synthesis. This figure also shows that application of heat or chemical to exotoxin can result in the deactivation of exotoxin. The deactivated exotoxins are called toxoid and that are not harmful to somatic cells. Exotoxin and cytotoxins kill or damage the host cell through membrane, neurotoxin, enterotoxin, superantigen and toxide. Membrane disrupting toxin lyses the host cell by making protein channel in the plasma membrane and disrupting phospholipid bilayer. Neurotoxin interfere with nerve cells. Enterotoxin affect lining of gastrointestinal tract. Superantigen overstimulating the body immune response due to the release of cytokines from the host cell. Toxide toxin that has been altered by heat or chemicals. Now, the last step of mechanism of pathogenicity is a portal of exit, where microbes leave the body and spread through the population. Syringe provided a portal of exit of microbes in the blood. Three common portal of exits are respiratory tract via coughing or sneezing, gastrointestinal tract via saliva or feces, and genito or urinary tract via secretion from the vagina or penis. Now, the identification of microbial pathogenicity. The typically identification is done by the growing the organism in a wide variety of culture, which can take up to 48 hours. The growth is then external visually or internal as a genomically identified. The culture organism is then subjected to various biochemical assays to absorb the reaction to helpful further identify species and strain. Sign and symptoms of pathogenic microbes. Each infectious disease has its own specific sign and symptoms. General signs and symptoms common to a number of infectious diseases include fever, diarrhea, fatigue, muscle sickening, coughing, sneezing and respiratory tracts. Treatment. Bacterial infection may be treated with antibiotic. They are classified as bactericidal if they kill bacteria. There are many types of antibiotics and each class inhibit a process that is different in the pathogens from that found in the host cell. Antibiotics are used both in treating human diseases and in intensive farming to promote animal growth. Phage therapy using bacteriophage can also be used to treat certain bacterial infection. Prevention measure. Alongside maintaining good general health, a person can take certain steps to reduce their risk of infection from pathogens. This includes hand washing and drying on a regular basis. Washing the hand regularly is crucial in helping to stop the spread of diseases. People should wash them at least 20 seconds with soap and water or an alcohol based sanitizer. It is particularly important to do this after doing outside activity, touching animals, going to the toilet, looking after sick people and sneezing or coughing. Second, wear mask while sneezing and coughing, consume only cooked food, avoid close contact with people who are infected. Keep your hand and finger away from your eyes, nose and mouth, maintaining social distancing. Stay at home when sick. People with infections should avoid going to school or work or having close contact with others to reduce the chance of spreading pathogens. Regularly clean and disinfect workplace. Contaminated surface or object can transfer diseases. Staying up to date on recommended vaccination. Vaccine against infectious diseases such as influenza, whooping cough, measles are available for babies, children and adults. Vaccine stimulate the immune system to recognize pathogens, destroy them and prevent future infection. Getting medical advice. Person can talk to a medical professional if their sign of infectious or treatment is not working. Summary. Pathogen which include bacteria, fungi, viruses, worm and even infectious protein are known as prawns. They are the source of infectious diseases. All classes of pathogens need to have way to get inside their host and avoid being instantly destroyed by the host immune system. The majority of microorganisms are not harmful. The pathogens that are comprises distinct virulence genes that facilitate interaction with the host, triggering specific reactions from the host cell that aid in the pathogen proliferation and dissemination. During an infection, pathogenic fungi, protozoa, eukaryotic parasites commonly go through a multiple form. The capacity of transition between various forms is normally necessary for the parasites to be able to remain in a host and cause diseases. Now, the topic to be covered in next lecture is on bacterial morphology. In bacterial morphology, we learn about the morphology and shape of a bacteria cell is the most distinguished property of bacteria. Morphological trait of a bacteria cell are an important factor in its ability, acceptability and evolution. There are many features like motility, mode of nutrition that are affected by the shape of bacteria. These are the references: Pelzar, Prescott, Tortora. Thank you. I hope student will understand this interesting topic, pathogenic microbes, well.